So given that, it's pretty impressive how accurate he was with his results. What he said was, every organism has two pieces of information, which we'll call alleles. They can have the same allele, they got them from their parents, or they can have different alleles. An organism that has the same alleles for some trait is called homozygous. It'll either be homozygous dominant or homozygous recessive. If they have differing alleles for that trait, we'll call them heterozygous. There's only one kind of heterozygous, that is that you have one of each kind of allele. So heterozygous, recessive, we don't say that. Now, here we have our two P generation flowers, the one with the white petals and the one with the purple petals. They both can only give one kind of allele to their offspring. So here's a lowercase p from one parent, uppercase P from the other, and this creates our heterozygous F1 generation. Now, because they have the dominant allele here, they're going to resemble their homozygous dominant parent. But we now know that if we breed them together, that the two of these heterozygotes together, we are going to get a variety of different offspring. Now here, where I was giving you examples for genotypes, I used the letters Q. In general, it does not matter what letter you use for your genotypes. You can choose whatever letter you want unless I specifically give you a letter for a problem. But if you're in a situation where you are allowed to select what letter you're going to use, I would caution you not to use certain letters. For example, if you have a capital X and a lowercase x, they look pretty darn similar to each other. Capital C and lowercase c look pretty darn similar to each other. I would suggest something like, for example, Q. Q, I'm never going to confuse your upper and lowercase from. A, I'm never going to confuse your upper and lowercase letters. So if I cannot tell, if you use like, some people do like, oh, this is my capital S and this is my lowercase s, I can't really tell the difference between these two. I'll just assume that you are incorrect if I can't work it out for myself. So make sure that you select letters where it's very clear what your upper and lowercase letters are. We now know that the physical trait that accounts for Gregor's rules are the chromosomes. That's the molecule that's essentially carrying this information that he was talking about. That the genes are located on specific positions on the chromosomes, which we call loci. And that because of how these chromosomes behave during meiosis, how they separate during metaphase 1, that is what actually accounts for the statistical results we see in these types of experiments. We refer to this as the law of segregation. The alleles are separated so that each gamete is only going to carry one copy. We talked about this towards the end of Unit 4. Every parent has two copies of each chromosome because they are diploid organisms, but then they make haploid gametes. Haploid gametes have only one copy of each chromosome. Each chromosome has one allele for each kind of gene. So instead of having two genes, you are going to create gametes with only that one copy. Let me draw it out here a little bit more clearly here. If I have a... So if I have a parent that has two differing copies of an allele, then they can either create, we'll say, a sperm cell with a capital P, or they can create sperm cells with a lowercase p. But this is a diploid organism with two copies of everything. These are haploid gametes that have only one copy of information. So here is some set of chromosomes that you have inside your cells. We got one chromosome from mom, one chromosome from dad. On each of these homologous chromosomes, at a certain point, we'll have a locus for some gene here. This is a gene for flower color in this instance. Now, it's going to be the same kind of gene in both locations, but the exact sequence of nucleotides might be different for them. And if you change the sequence of nucleotides, you change the resulting enzyme that is synthesized from that gene. If you have even one functional copy of this gene, you are going to make the purple color pigment that results in purple flower color. If you have two broken copies of this gene, two copies that do not create this enzyme, then you will create white pigment flowers. But as it turns out, whether you have one or two copies, it doesn't actually change how much of the pigment is produced. As long as you have one copy, you will produce pretty much the same amount of pigment, and you'll end up with purple flowers. And this is why purple is dominant to white in this particular instance, because if you have one copy or two copies of the purple flower gene, you get purple flowers. Only when you have two copies, two alleles for the trait of white flowers, will you actually fail to make any kind of pigment, and then you will see white flowers. Now, some dominant genes are like that. It's just because you need one functional copy of the enzyme or not. Others are dominant recessive because there's some sort of chemical interaction between the genes. They are able to kind of switch each other on and off, and it's a little bit more complicated. The idea of a dominant and recessive gene is really just a simplification of a couple of different effects that we might see. And not every gene follows this pattern. We'll see some exceptions in a little bit. So, Mendel realized that because the only thing he transferred from one plant to another was the pollen, that the parents' alleles have to separate during the production of gametes, that's what's actually carrying this information to the next generation. So, without knowing about chromosomes, without knowing about DNA, he was mathematically able to realize what we learned in Unit 4, that diploid parent cells become haploid gamete cells, that you go from having two copies of every chromosome to having one copy of every chromosome. And these gametes that you make at the end, they're only going to have one of each of the parent's alleles for any given gene. Let's do an example genetics problem here. So here's a typical question that I might actually give you on an exam or as homework or something along these lines. We have a father and a mother, and they're going to breed together. They're going to create some offspring between them. They have genes for what's called the Rh blood type. Rh stands for the rhesus factor because it was initially found in rhesus monkeys, but it turns out it exists in humans too. If you get a blood test and they'll say you're either O positive or O negative or something like that, the Rh factor is this positive negative right here. It turns out it's totally separate genetically from the AB, or O blood types. So you can either be positive or negative, and this exhibits a simple dominant recessive behavior. The positive blood type is a dominant trait. Here we have the father's genotype and phenotype listed. Okay. The genotype is what alleles he has. He's a heterozygous individual. He's got one of each kind of allele, dominant and recessive. Because he has a dominant allele, he has also the dominant phenotype here. So that is his situation. Now, the mother, I have not listed what her genotype is, but I have told you that the phenotype is negative. But because we know that the positive trait is the dominant trait, and she must therefore have the recessive phenotype, we know that she must have the homozygous recessive genotype. So let's make a Punnett square with these two. And first thing we'll do is we'll fill in the father's genotype up here. He's got a capital R and a lowercase r. Then we can figure out what the mother's genotype is by looking at her phenotype. She has the recessive phenotype, therefore she has the homozygous recessive genotype. If she had any positive alleles, she would have the positive blood type. That's what dominant means. So this is how we know that she must have 2 recessive alleles. Now, for each one of these squares is a 25% chance of an offspring of that kind. Okay? Every single time that these two have a kid, it's going to be another roll of the dice what combination of alleles they end up getting. So for this first square, we'll do the bottom left here. We'll take a look at the left-hand column for the father's alleles, the bottom row for the mother's alleles, and you get a heterozygote here. You get one allele from the father, one allele from the mother. Now, if you are a heterozygous, you end up with a positive blood type because positive is dominant in this situation. If we take a look at these alleles, it's also possible that they will produce an offspring with the homozygous recessive genotype, and that would be the negative phenotype. Now, if you take a look up top, well, we still have the father with 50% of his alleles are positive and 50% negative, 50% dominant, 50% recessive, and the mother can only give this same kind of allele. So it's actually going to be the same split up here in this top row as we had in the bottom row. And this is where I can reveal that these are not hypothetical parents. These are actually my parents. And my parents had 2 boys, one of which ended up with the negative blood type, that's me, and then the other one who ended up with the positive blood type, that is my brother. So if these two were to have another child, it would be another 50-50 possibility as to whether or not you were going to get a heterozygote or a homozygous recessive individual. So here you can start to see where Punnett squares are no longer just showing you that same 3-to-1 ratio. If we want to do the genotypic ratio here, the GRAT, for this Punnett square, we have 2 different combinations, 2 combinations where we have, I do it this way, a heterozygous offspring, 2 combinations where we have homozygous recessive. That's this one and this one here. For the phenotypic ratio, we have 2 positives to 2 negatives here. Now with ratios, you always reduce to the smallest terms, so both of these would become a 1-to-1 ratio here, because 2 and 2 are both divisible by 2, so you get 1-to-1 at the end. Now again, this is not a map of what offspring these parents are guaranteed to have. If they have 4 kids, they aren't definitely sure to get 50% positive and 50% negative. Every fertilization event is another set of coin flips. So if we were to imagine flipping 2 sets of coin, one a gold Sacagawea dollar and one a nickel-plated quarter, and we were to call heads the dominant allele and tails the recessive allele. In this case, we are mixing together 2 heterozygous individuals, so this is a monohybrid cross. And there is a 50% possibility that the first coin will end up heads, 50% possibility that the second coin will end up heads. This means that overall, 1/2 times 1/2, you will get 25% chance that we're going to get 2 heads in a row, 2 dominant alleles. You have 2 different ways where we can have 1 head and 1 tails. So there's actually a 50% possibility of heterozygous. Right? So approximately half of the offspring from any 2 parents like this are going to have the heterozygous genotype. And there's 25% odds as well that they're going to get 2 recessive alleles. Okay, so if I were to say, these parents had 30 kids, all of whom somehow ended up with the dominant genotype, with the homozygous dominant genotype, what are the odds that their next kid is going to have homozygous dominant? The odds are still 25%, because for every kid, it's another flip of the coins, another 25% possibility here. The number of kids that you've had before this do not matter. Using what we know so far, we're going to experiment with something called a test cross. Now, here's what information you're going to have at the beginning of this problem. Dog coat color is controlled by a few different genes, but for right now we're going to say it's controlled by one gene. We're going to use the letter B for the coat color gene, and it comes in 2 different alleles. Capital B is black fur. Lowercase b is chocolate fur. So we want to know what the genotypes of these 2 dogs are. Their phenotypes are self-evident. This is a black dog. This is a chocolate dog. Right? Those are their phenotypes right there. But their genotypes, that takes a little bit of understanding in order to arrive at. Black fur is dominant to chocolate fur. So for this dog to have chocolate fur, that means that she must have at least 1 chocolate allele, right? But what's the other allele? Well, if she had a dominant allele, she would have black fur, because that's what dominant means. So she must have 2 recessive alleles. If you have the recessive phenotype, chocolate in this case, you must have the homozygous recessive genotype. The black dog, on the other hand, we know that since he has black fur, he must have at least 1 black-furred allele. However, we don't know what the second allele is, because it could be that he has 2 black alleles. It could be that he's heterozygous. Both of these would result in the same appearance of the dog. So how do we know which one he is? Well, when you're solving a genetics problem, the first thing you do is write down everything you know on the surface of it. Here's what we know. Black is dominant to brown. Black is dominant to chocolate. The mother has 2 recessive alleles. The father has at least 1 dominant allele, but we don't know what the second one is. Once you have all the information that you know absolutely written down, you start to look between generations. So if we look at the puppies that these 2 parents create, we can get a little bit more information. Okay? If this second allele is a dominant allele, well, then there's only one kind of allele that he can pass on to his puppies. And if all of the puppies get a dominant allele, they all are going to end up with black fur. If, on the other hand, this second allele happens to be a recessive allele, well, we already know that the mother is going to give all of the offspring a recessive allele here. So if the father can give 50% of his offspring a recessive allele, that means that 50% of the offspring are going to be chocolate and 50% are going to have black fur. Here, I'll do the full Punnett squares just to drive this point home. So we have 2 possibilities. We know what the mother's alleles are, no matter what. The mother has 2 recessive alleles. She's homozygous recessive. That's her genotype. The father can be one of 2 different genotypes. He can either be homozygous dominant or heterozygous. If he is homozygous dominant, that means that every single one of his offspring must get a dominant allele, and the mother can also only give 1 kind of allele. Every single one of these puppies has black fur. Okay. Black fur is the phenotype. Heterozygous is the genotype. For the phenotypic ratio, we have 4 black-furred puppies to 0 of anything else. That reduces down to a 1 to 0 phenotypic ratio. For the genotypic ratio, we have 4 heterozygotes to 0 of anything else. That reduces down to a 1 to 0 genotypic ratio. So in this case, the two ratios will be the same. Down here, only 50% of the puppies get the dominant allele, but all of them get at least one recessive allele. 2 of them are going to get 2 recessive alleles. So we are going to have 2 different phenotypes on display with these puppies. Black and chocolate puppies. The phenotypic ratio here is 2 black-furred puppies to 2 chocolate puppies. That's what we call a 1-to-1 phenotypic ratio. The genotypic ratio is going to be 2 heterozygotes to 2 homozygous recessive, which is also a 1-to-1 ratio. So some students get a little bit tripped up with the Punnett squares. They think that it's always going to turn out to be a phenotypic ratio of 3-to-1 and a genotypic ratio of 1-to-2-to-1, but it really depends what the inputs of the parents are. You can get all kinds of different ratios depending on what the specific alleles of the parents are at the end. Notice, 1-to-0 is not the same as 1-to-1. We can get different results.